Okay, thank you, Deanna. Well, Molly, it's an exciting day. You have two departments worth of people who are going to miss your calm confidence and your smiling face at Lawrence. The biology department, we have been gifted with your presence in the stockroom there and that magical team of people who seem to manage to get stuff done with remarkable efficiency. So you've been a force there, but you've also been a force on the second floor of chemistry in the biochem lab, where you've worked tirelessly on this project for quite a few hours. In the past 2 years, you've impressed many people with your independence and your perseverance that you've shown in your research and all of your academic work. Your science is thoughtful, carefully executed, and well-informed by literature. And when you need it, you seek and really welcome, receive well feedback about your work in order to move forward. All of these attributes are going to serve you well as you embark in your life after Lawrence. It's really been a pleasure working with you especially this past year, and helping you when needed, which has been actually relatively little. And so congratulations, and I'm really looking forward to hearing about your low complexity regions of yeast Ataxin-2 today.
Alrighty, my project is modeling neurodegenerative disease, low complexity regions, and phase separation of yeast Ataxin-2. And you've now met my advisor, Kim Dixon. Thank you so much for the introduction, Kim. Right, this is where we find out that my mouse doesn't work. There we go. All right, so we start today with the big picture item, neurodegenerative diseases. Sadly, most of you watching this presentation probably know someone who has been impacted by a neurodegenerative disease. These diseases are characterized by the degradation of neurons or nerve cells. There's a drawing of one to the right here. This often leads to movement disturbances called ataxias and cognitive impairments called dementias. Three of the most common forms of neurodegenerative diseases are Alzheimer's disease, Parkinson's disease, and the focus of our presentation today, ALS. Amyotrophic lateral sclerosis, better known as ALS or Lou Gehrig's disease, causes the death of motor neurons, which are the nerve cells that allow your muscles to move. The death of these cells leads to worsening paralyses. Patients may lose the ability to speak, use their hands, and walk. Toward the end of life, this begins to affect respiratory muscles too. Most deaths from ALS are due to respiratory failure. Unfortunately, there are very few treatment options for ALS at the moment, and there is no cure. In order to treat or cure a disease, you often need at least a baseline understanding of what causes the disease. Unfortunately, we don't really have that for ALS, but scientists have some general ideas that they're trying to piece together. There are many, many avenues that scientists are currently investigating, but we'll look at three today in the context of my research and some previous research that's been done. The first avenue is genetics, or the DNA blueprint that tells cells how to make proteins. There are some genetic anomalies that make people more likely to develop ALS. The second avenue is mitochondrial dysfunction. Now, we all know that the mitochondria are the powerhouse of a cell. For those of you who don't do memes, mitochondria make ATP, which is the energy currency of the cell. If the mitochondria are dysfunctional, the cell is probably going to struggle and potentially will die. Mitochondrial dysfunction is implicated in several neurodegenerative diseases, including ALS. The third and final avenue we'll look at is aberrant intracellular communication. This means dysfunctional or confused communications between the components inside of a cell. For example, if some other cell components start giving the mitochondria confusing messages, the mitochondria isn't going to understand and may act in unhelpful or dysfunctional ways. It's a little like if your coworker started emailing you only in wingdings. Now, scientists are trying to fit these three concepts together to explain why some people get ALS. And one explanation that links these three things together has to do with a protein, and that protein is called Ataxin-2. Ataxin-2 is a human protein that is involved in several different cellular processes, including processes that have to do with cellular metabolism. But what's most interesting about Ataxin-2 is its association with ALS. Ataxin-2, like all proteins, is coded for in your genes. And when these genes are normal, the protein is normal, and it functions normally. However, mutations can occur that affect the structure of Ataxin-2. And previous studies have shown that these mutations are associated with an increased risk of developing ALS. Now, these mutations exist in specific areas of the Ataxin-2 protein. And these areas are called low complexity regions, or LCRs. Now, I'd love to give you an easy definition of an LCR right here, but in order to understand what an LCR is, first we need to understand how Ataxin-2 and all proteins are made inside of the cell. So in order to understand that, we'll start with something called the central dogma of molecular biology. The central dogma says that your DNA is first read and converted into RNA through a process called transcription. Then the RNA is converted into a string of amino acids called a polypeptide. The circles here represent those amino acids, or the chemical building blocks of a protein. However, this is not the end of protein synthesis. In order to make a fully functional protein, such as Ataxin-2, you need to give this polypeptide chain a 3D structure. In order to get this shape, the polypeptide chain must go through processes called protein folding. So when you start the protein folding process, what you have is that series of amino acids all strung together into a polypeptide chain. This is sometimes likened to beads on a string. But what you ultimately want is for those beads, those amino acids, to fold and twist around one another, making a globular shape. The final shape of the protein is really important because how a protein folds determines its function or dysfunction inside of your cells. This folding is influenced by many, many different things inside your cells. But one of the most important is the different personalities of the amino acids. Since each amino acid is a different chemical, it will react differently to different things. Some amino acids love water, some amino acids hate water, some amino acids have a positive electrical charge, a negative electrical charge, or no charge at all. During protein folding, for example, amino acids with a positive charge might be attracted to amino acids with a negative charge, and they might stick together sort of like a magnet. And amino acids that really hate water are going to try to be on the inside of the protein where they're not as likely to have to interact with water. So what amino acids are present tell us something about how a protein is going to fold, and thus how a protein will eventually behave. And this finally brings us back to those low complexity regions, or LCRs. LCRs are regions of protein that have repeated amino acids. That is, they have tons of the same amino acid in the same region. If you look at the drawing on the right here, you see that there are 4 types of amino acids in this example of a polypeptide chain, but that middle section with the bracket over it only has 1 type. That section is an LCR. Now, we said before that amino acid personalities determine how a protein folds. Well, the thing about LCRs is that they kind of lack a diversity of personality. It's the same thing over and over again. And this makes protein folding, well, a little wonky. Because of the lack of diverse personalities, these regions typically lack a set or given 3D structure. So they're sometimes called floppy regions. However, this isn't necessarily a bad thing. Proteins have LCRs for a reason. Because these regions aren't folded up into a tight globular shape, they're really easily accessed by other chemicals that are floating around inside of the cell. This allows them to act as cellular sensors. For example, if the pH of a cell suddenly drops, that's probably a sign that something is going really, really wrong inside of that cell. And because these LCRs are more easily accessed by the chemicals that caused that pH to drop, the LCRs might be able to sense it and start sending out warning messages to other parts of the cell. But this only works if the LCR is coded for properly. Mutations in an LCR can mean real trouble for that protein and that cell because it might mean that it's acting as a poor sensor or poor communicator. Ataxin-2 has an LCR known as the poly-Q LCR. This means that it has a ton of the amino acid glutamine, which scientists have given the 1-letter code Q. The glutamines, or Qs, are shown here in purple. When the poly-Q region is normal, Ataxin-2 functions normally. But there's a specific genetic variant that codes for an expansion of the poly-Q region. This expansion is associated with two neurodegenerative diseases, one of them being ALS. This is one of the most common mutations in cases of non-familial ALS, and some therapeutics are actually being tested in mice that target this LCR. Now, interestingly, Ataxin-2 has another LCR that's decidedly less famous and not as well studied, and that's the methionine-rich LCR. Just like the poly-Q LCR had a lot of glutamines, the methionine-rich LCR has a lot of the amino acid, you guessed it, methionine. Some research is suggesting that these two regions might be talking to one another. Specifically, they might be passing on information about mitochondrial function. Normally, this communication is healthy. The mitochondria signals to the methionine-rich LCR that something is going wrong. The methionine-rich LCR gets the message and passes it along to the poly-Q LCR, and this leads to cellular signaling and an induction of cellular activities that protect the mitochondria and other cell components from whatever is causing the stress. But what if there's an expansion of that poly-Q region? Well, this might spell some trouble. The mitochondria and the methionine-rich LCR might communicate just fine, but if Ataxin-2 has an aberrant poly-Q region, it might start sending some messages that the other LCR and the mitochondria ultimately misinterpret or just don't understand in the first place. This might leave the mitochondria and other cell components without protection from the stressor. This could get even worse if there is also a mutation that affects the methionine-rich LCR too. This could lead to dramatically confused intracellular communication and leave cell components, such as the mitochondria, without help and without protection. Now, this explanation ties together our first three components really neatly, but obviously in biology, just because something makes sense doesn't mean that that's what's actually happening. We need to test this hypothesis, but that means we need something to test it in. Now, we definitely can't do this in humans because we can't ethically induce something that could cause a disease. So can we do it in human cells? Or what about non-human animal models? The answer to that is maybe, even probably, but those models are finicky. Human cells and animal models take a lot of time and energy to upkeep. Not to mention these models are also expensive. And in the case of animal models especially, it has ethical concerns. This is early research, and it's worth testing this model out on an easier, cheaper, and less ethically fraught model first. So, are there better models? Well, I think so. And the authors of the papers that I studied this summer say, yeah, there is. Yeast. Saccharomyces cerevisiae, also known as baker's yeast or brewer's yeast, is a great model organism for studying human diseases. This is because yeast are easy to maintain and manipulate, far easier than human cells or animal models. They are also cheap and readily available, at least before a pandemic that caused a lot of people to stress bake. And they are eukaryotic, which means they have membrane-bound organelles like human cells. These qualities make them a compelling model organism for studying diseases, and it appears many labs agree with me. This past summer, my research was based on two papers that were published in Cell in April of 2019. That's right, we do cutting-edge research here at Lawrence University. These papers used yeast models to investigate Ataxin-2. I'm not going to read the titles to you because they are even more of a mouthful than the title of my presentation. However, they were specifically investigating how that methionine-rich LCR of yeast Ataxin-2 communicates with the mitochondria. Notice that I said yeast Ataxin-2. That's because, technically, yeast don't have Ataxin-2. Rather, they have an Ataxin-2 ortholog. Now, orthologs are kind of messy and complicated to explain, so we're going to move forward with the assumption that ortholog means yeast version. That is, the human protein Ataxin-2 has a yeast version of it. Orthologs are more complicated than that, and there's some biologists in the wings who are a little bit stressed out right now, but we'll move forward. The yeast version of Ataxin-2 is called PBP1. It has many of the same functions as Ataxin-2. For example, it is still very much involved in cellular metabolism, but critically, it also has LCRs, one of which is also methionine-rich. So the researchers of those two papers were looking at the methionine-rich LCR of PBP1. I've shown it to you in cartoony form before, as here. But for those of you who are interested in into what the actual methionine-rich LCR of PBP1 looks like, it looks like this, where each of those blue M's is a methionine. The researchers were specifically looking at how this methionine-rich LCR communicates. And that has to do with a question that some of you may have had sitting in the back of your heads for a little bit now. Just how do proteins and LCRs talk? Aren't they parts of protein? Proteins and mitochondria can't talk. Well, they can and they do in a sense, just not obviously how we talk. And if we want to understand how they're talking, we need to get a better picture of their language. And the language of PBP1 and many other types of proteins is a process called phase separation, specifically liquid-liquid phase separation, which is when two liquids are present together but are not fully mixed. Interestingly, you've probably seen liquid-liquid phase separation before but didn't know it. The behavior of oil and water can be thought of as a type of liquid-liquid phase separation, as well as separated paints. The image on the right here is by an artist who uses different liquids to demonstrate that what phase separation looks like, because, well, it's super cool. But it's also now being recognized as a hugely important concept in biochemistry, specifically in protein biochemistry, because many proteins have the ability to phase separate inside the cell. And this includes our protein of interest, PBP1. Now, protein phase separation can be functional, so helpful, or aberrant, which is unhelpful. Functional phase separation occurs when there's a stressor. The level of phase separation is congruent with the severity of the stressor, and those phase-separated droplets disassemble when the cell tells them to. This allows the cell to sequester proteins, giving them control over cell activities and helps protect components of the cell from whatever the stressor is. Aberrant phase separation occurs when the level of response is not congruent with the severity of the stressor, and the droplets either persist when the cell is telling them to break up, or when the droplets don't form at all, even when the cell needs them to form. This leads to loss of control and possibly disease. Like we said before, PBP1 phase separates, and this is functional, usually. Those two papers I studied found that functional PBP1 phase separation helps to regulate the mitochondria. Specifically, phase separation inhibits TORC1. TORC1 function is a little bit complicated, so for this presentation, what's important is that you know it is also a mitochondrial regulator. So ultimately, PBP1 phase separation is contributing to a chain of communication that tells the mitochondria how to respond to changing cellular environments. And that's important. We want our cells to detect issues and respond to them and not stay static. But how does PBP1 detect things? The authors of the papers found that it does so through its methionine-rich LCR. Specifically, the researchers found that there were 8 methionines that were critical to proper phase separation abilities. In this figure here, which is from one of the papers, we're looking at the methionine-rich LCR of PBP1. The angled black letters are the methionine positions, so where in the LCR these methionines were located. WT stands for wild type, or the literature sequence for this LCR, and below that you see 2 experimental strains, MS8A and MY8A. The researchers induced mutations in both strains and then looked at the effect on the phase-separated droplets. In MS8A, they converted those 8 critical methionines to the amino acid serine. This resulted in destabilized phase-separated droplets. In MY8A, they converted the 8 critical methionines to the amino acid tyrosine, shown here as the green Ys. This resulted in highly stable phase-separated droplets. Now, yes, those are 2 completely opposite things, but either of these eventualities could be very bad, because what it ultimately means is that PBP1 has lost its responsiveness to the cellular environment. That's bad news if you're in the sensing and communication game, as PBP1's methionine-rich LCR appears to be. So what do we ultimately gather from those 2 papers? First, we gather that PBP1 is the yeast ortholog of human Ataxin-2, and it phase separates. Second, we learned that the methionines of the methionine-rich LCR of PBP1, specifically 8 of them, facilitate proper phase separation of PBP1. Third, we know that when PBP1 phase separates, it helps regulate mitochondrial functioning. And last, we know that proper mitochondrial regulation is critical for cellular health, and mitochondrial dysregulation is implicated in several forms of neurodegenerative diseases. Okay, so we've made it this far. You've got through all of the background. Now we have enough background information to discuss what I did this summer. So this was the question that I had at the start of summer 2019. Do natural differences in the amino acid sequence of the methionine-rich LCR of PBP1 lead to differences in mitochondrial functioning? So how did we actually go about getting that go about answering that question? Well, first I obtained some yeast strains from Vincent's Meyer. Thanks, Vince. Then I grew them in one of two conditions. Half were grown at room temperature and half at heat shock conditions, or 35 degrees Celsius. What's important to know about heat shock is that yeast do not like it. Specifically, their mitochondria do not like it, and they get stressed out. This is a good way then of looking at the effect of mitochondrial stress on the yeast. Then I looked at the amino acid sequences, which tells us about the differences between the strains. And then I looked at something called the mitochondrial membrane potential, which tells us about mitochondrial function. So to go into more detail, let's start with the amino acid sequencing. First, I did something called PCR, which stands for polymerase chain reaction. It can also stand for pipette cryo-repeat if you've had to do it one too many times. PCR is important because DNA is fairly small. If you want to study a small region of DNA and you just try to do it with the DNA that you naturally have, your tests are never going to be able to see it. So you have to amplify it, which means copying the same specific region of DNA over and over again until you have enough that your tests, sometimes called assays, can see. And so I did PCR on that methionine-rich LCR of PBP1. Then you put all of your work into a single box, wrap it in as much styrofoam bubble wrap and packing tape as you can, say a prayer that it doesn't get damaged, and ship it to a company that can do more advanced sequencing than Lawrence can. Then the sequence data comes back, and what you get are the nucleotide sequences, or the letters that code for the amino acid sequences. So we don't have our amino acid sequence yet, we have our DNA code. Now we need to decode it so we can get our amino acid sequences. And luckily, the NIH actually has a free online database that allows you to do just that. So you plug in your sequence, all those letters, and you get an amino acid output that allows you to look for mutations or changes in those amino acids. And these were the mutations that we saw. There were only 4 positions that had amino acid mutations, and that's consistent with some previous research that found that this is a highly conserved region. That means that mutations are relatively uncommon even across many generations. So let's walk through how to interpret this figure. The very top row is the position, so the relative location of these mutations. Reference means the accepted literature sequence for this region. And those numbers below reference, those are our different strains of yeast. So 63 and 1335 are 2 different strains, and 63 cross 1335 is their offspring. Now let's look at a specific mutation. At position 20, you see that there should be a methionine. In strain 63, you see an isoleucine instead. So that's a mutation. And I was really excited about that mutation too, because it was a methionine, which is obviously really important. However, some differences occurred. Hold on one second, I'm having some screen share difficulties. We'll be back in one moment. So the idea here is that we were looking for those amino acid mutations, and we have a mutation of that methionine that I was very excited about. However, I'm sorry, this is not going well at the moment. There we are. All right. So we've gone through, we had that mutation. Right here is our amino acid 591, which is that first amino acid that was considered critical in the, so it's that first of the 8 amino acids considered critical. And here's my amino acid where I had the mutation. So I literally missed it by a singular amino acid. So unfortunately, I did not see any mutations in any of the amino acids that were considered critical to proper phase separation. So that was a little disappointing. However, this is really messed up. There we go. Hi all, sorry. We'll be back on board in just a second.
Anything I can do to help, or are you okay?
I'm okay. I just, all of a sudden, my computer decided to not cooperate with me. So I will be back up in 2 shakes of a lamb's tail. All right, we're officially back this time. So to go back over
You're not
what I was
sharing.
saying there,
You're not sharing.
Not sharing, of course we're not. Okay. Are we
now you're sharing. Now you're sharing.
Beautiful.
And you're cut off.
Am I cut off? There we go.
There you go.
All right, now we're officially back. So what I was saying is that unfortunately, the amino acid mutation that I saw, so that methionine mutation, was not one of our critical methionines. So there's the first critical methionine, and there's mine. And what added insult to injury is that if you look at the slanted numbers on top, you'll recognize that some of those methionines had other amino acids in between them. That is, like you go from 585 to 587, which means that there were other amino acids between there that aren't shown in this. But if you look at 590 and 591, you see that they're right next door to each other, which means I missed it by 1 amino acid. So all is not lost, though. The data are data. Data are data. So to conclude this mini section, our first conclusion is that there were a relatively low number of mutations, which corroborates previous research that showed that this region is highly conserved, which could suggest that it has important function because it is highly conserved. DNA necessary for life tend to be highly conserved regions. And this leads to the question, if natural variations in this region are so uncommon, are they likely a culprit for neurodegeneration? Well, maybe or maybe not. We'll get back to that in a little bit. So the next portion that we go to is mitochondrial membrane potential. Oh, yep, that's why that was our error. My slide got mixed up. Okay, so does the rest of this matter? Maybe. There were mutations, and one of them was a methionine. It just wasn't one of the 8 critical ones. So let's take a look if any of the other mutations have any effect. And we need to go to mitochondrial membrane potential in order to do this. Okay, and remember, mitochondrial membrane potential gives us an idea of mitochondrial functioning. So to go back a slide, because this is where my thing got all messed up, mitochondrial membrane potential is basically a measurement of the proton concentration outside of the membrane versus the proton concentration on the inside of the membrane. The protons are shown here as orange dots with the H+ inside. And the mitochondria use the proton gradient to drive metabolic reactions, so to generate ATP. And so mitochondrial membrane potential, which again is the electrochemical difference from one side of the membrane to the other, is important and tells us about the mitochondrial functioning. Okay, so mitochondrial membrane potential, how do we measure it? Well, we do so through something called flow cytometry, specifically fluorescent flow cytometry. And so the first thing that you do in flow cytometry is you tag the mitochondria with a fluorescent dye known as RH123 or rhodamine 123. It enters the mitochondria and causes the mitochondria to fluoresce. And then you can go over to the flow cytometer, which is a machine that basically takes your cells and blasts them past the sensor one at a time with a really high pressure, and your output is your level of fluorescence. All right, so you're looking at individual cells that have a fluorescent tag in their mitochondria, you're blasting them past the sensor, and you're seeing how much they're fluorescing. So the output is your level of fluorescence, and low levels of fluorescence mean high mitochondrial membrane potential, and high levels of fluorescence mean low mitochondrial membrane potential. To give you an idea of exactly what that output looks like, we have something called a histogram on the left here. This histogram is looking at geometric mean of fluorescence. Again, if you have a high geometric mean of fluorescence, you're looking at low mitochondrial membrane potential and vice versa. Now, to make this a little bit more complex, we also added a chemical called guanidine hydrochloride. Now, guanidine hydrochloride breaks up phase-separated droplets, but doesn't itself affect the mitochondria. To summarize, we're looking at two different types of yeast samples that were passed through the flow cytometer. Some were grown without 5 millimolars of guanidine hydrochloride, and some were grown with 5 millimolars of guanidine hydrochloride. Without leaves the phase-separated droplets intact, and with the guanidine, the phase-separated droplets are broken up. And this lets us to compare mitochondrial membrane potentials with and without phase separation. Okay, so now we have more data, this time from our flow cytometer. This is a graph on the y-axis of the geometric mean of fluorescence, and on the x-axis is our different strains of yeast. So let's take you step by step through this. The blue shades are grown at room temperature. So these were our non-heat shocked yeast. The purple strains are heat shocked yeast, so stressed yeast, effectively. The dark colors, so dark blue and dark purple, represent our strains grown without guanidine, which means that they have their phase-separated droplets intact. And then the light colors are grown with guanidine, which means they have their phase-separated droplets broken up. Now, it's important to note here that what we're looking at is we don't want to look at 63 versus 1335 because sometimes yeast just have different mitochondrial functions. Vince can tell you all about that next year, I'm sure, in his senior experience project. And so what we're really actually interested in is, if my cursor will let me, is this difference here. So between 63 without guanidine and 63 with. So this difference right here. And remember, we have to interpret this a little bit backwards, where our room temperature 63 without guanidine actually has higher mitochondrial functioning than 63 with guanidine. All right, so now we've seen this graph. The graph on the next slide is the same thing, but now it's that percent change, right? So if we look here, we're on that first blue bar is going to be this, but in percent change form. So that's what we're looking at here. And the way to interpret this for 63 is that there was a huge positive change, right, between the with and without guanidine. And what this suggests is that there's lower mitochondrial functioning when you break up those phase-separated droplets. So this is our room temperature. Now you'll notice that there's nothing here for heat shocked, and that's because that difference was about -2%. So the difference basically disappeared when you heat shocked them. And that was really compelling to me because remember, 63 has that mutation in it. It has a mutation in the methionine that's right next door to our critical methionines. And so I was very excited about this because what it would suggest is that the phase-separated droplets are advantageous for mitochondrial functioning at normal situations, but then when you heat shock them, all of a sudden, phase separation seems to maybe become somewhat aberrant. That's interesting. And I thought it was interesting until I looked more closely at a couple of our other strains. So strain 590 has the, so excuse me, so 63 here is functioning very differently than 1335 cross 63 cross 1335. Remember, we have a mutation here. Now, 590 has the exact same mutation. And if you look at it, it has wildly different function. So all of a sudden you had, in 63, you had this gigantic change when you heat shocked it, right? And that pretty much disappears in 590, but the amino acid mutation is exactly the same. So what did that lead me to conclude? Well, that amino acid sequence probably wasn't the only thing that was contributing to phase separation behavior in these yeast. To further confirm that, I looked at 3 strains, 1335, 63 cross 1335, and 1198 cross 218, which all have the same amino acid sequences and wildly different mitochondrial functioning, right? So you can see here 1335 has fairly low mitochondrial functioning when heat shocked, and a negative value actually suggests that the mitochondrial functioning was higher when you broke up those phase-separated droplets. So the basic takeaway here is that there really was no correlation between the amino acid sequence and the mitochondrial functioning. So to get to our conclusions then, what did we actually draw from this data? Well, first, that the mutations of those 8 critical methionines appear relatively uncommon. Now, that it's hard to say that they would not occur naturally, just that they don't appear super common. And in order to state that definitively, we would have had to look at way, way, way more strains. We would have had to look at tons of them in order to say this definitively, but at least from what I did, mutations of those 8 critical methionines from those 2 papers don't seem to be very common. Additionally, the amino acid sequence of the LCR did not appear to independently affect the mitochondrial membrane potential of the yeast, which means that additional factors may be affecting phase separation. So am I saying that a paper published in Cell is wrong and that I was just able to uncover all of the secrets of the yeast universe? Well, no. There were relevant differences between these studies. Those 2 papers that I referenced earlier, well, they were seeking to understand behavior of lab strains of yeast, and I was using wild and brewing yeast. This is important because lab strains of yeast tend to be coddled strains. They really don't react well to stress. But wild and brewing yeast are quite hardy and have to be exposed to stress quite regularly. There's a reason you might want to use wild and brewing yeast to model human disease, because our cells are exposed to stress all the time. We drink too much coffee, we stay up too late, and we have stressful homework. Additionally, they created huge mutations, right? They mutated all of those 8 amino acids to a new amino acid. But I was using naturally occurring mutations that swapped out one or two amino acids here and there. And there's an argument to be made for my choice as well, right? Huge sweeping mutations are not likely to occur, not likely to occur naturally, especially in highly conserved regions. And so the question kind of becomes then, well, if you're creating massive mutations, do we really think that that's what's occurring in human cells to cause disease? Now, I also made some assumptions. The first assumption that I made was that guanidine hydrochloride would not independently affect mitochondrial membrane potential. Some of you may know guanidine hydrochloride is kind of a nasty chemical, though. So what if that wasn't true? Well, that would mean that the samples that I incubated with guanidine probably destroyed proteins more generally, which would just lead to differences in mitochondrial functioning regardless of that LCR. I also assumed that higher mitochondrial membrane potential was always good. And for our purposes in this assay, that was probably true. But there's some literature that would suggest that sometimes really high mitochondrial functioning or high mitochondrial membrane potential is going to actually be aberrant too. We also assumed that our assay would definitely show differences in mitochondrial functioning, but there's also a possibility that our assay just wasn't sensitive enough to really pick up changes. So to go back to, why do we care about these ideas? Well, we have those two LCRs in Ataxin-2. And that bottom region, that poly-Q LCR, is associated with increased risk of ALS. If these two LCRs are miscommunicating, we could be having aberrations in mitochondrial functioning, which again is linked to several types of neurodegeneration. So the final question is this cascade what's contributing to ALS? It may be, and the papers and my research don't say otherwise. But if we want to create a good model for this, we need to pay attention to what type of yeast models we're using and how likely the mutations are to actually occur in the natural wild type cells. So with that, I will go to my acknowledgements. There are many, many people that I would like to thank for their help with research. Too many of them to thank here, but the short list is my advisors, Kim Dixon and Deanna Donahue, for all of their help and advice. To the chem and the bio stockrooms, all of the staff, but most especially Joanne Stam, Eric Llewellyn, and Daniel Martin, for all of their help with getting reagents, getting access to equipment, etc., and specifically for DMART's SAGE safety advice, which is just don't be dumb. And then Lawrence University Summer Research for funding my research. And then I will go to questions.
Thank you so much, Molly. Are there any questions?
Oh, Kim has a question.
So Molly, what do you think the value might be of looking more deeply at the genetics of your different strains? I don't know how closely they're related, but I do wonder too that the Ataxin-2 isn't working in a bubble, and that there might be other things that even if specific genes to sequence without even needing to do whole genome sequencing. Do you have a sense of if that's worthwhile based on how similar or different the strains are and what genes you might target?
Yeah, I think to a certain degree. I don't know that I have any specific genes that I have in mind at the moment that I would target. Some of the original design of the research that didn't quite get borne out in the end was looking at differences more generally between parent and offspring. You can get some really interesting information about how yeast are passing on traits just through that. I think Vincennes is looking, at least to a certain degree, at some of that. So I agree. Just differences in other genes is probably going to be a big contributor. I was also only sequencing a really tiny portion of Ataxin, or excuse me, of PBP1. And so one of the things that if I was to speculate wildly, if I needed to get more funding, would be I would want to look at some of the other areas of Ataxin-2 and PBP1, specifically because there could be mutations as well in that poly-Q region. And so it's going to be, it's a matter of crosstalk, right? And so I would want to look, so they haven't identified a poly-Q region in PBP1. So the first thing is I would want to know is there a poly-Q region in PBP1? And were there mutations there such that could be contributing to poor communication between areas?
Cool, thank you.
Other questions? So this research project is quite diverse and you had to access a lot of different skill sets. What do you think the most challenging aspect was and what was the most rewarding aspect?
Yeah, so I think the most challenging was that at the beginning of the summer, I was trying to do a type of assay that would have looked for, yeah, Kim is laughing. I was to look for aggregation, so I would have actually been able to see if the proteins were clumping together. And that was done via Western blot, and that had worked all year long. And then summer started, and the Western blots just stopped working. So I had to eventually make the decision to move on from that type of assay as much as I wanted it to work. So I had to leave that behind. But then I would say that the most rewarding flow cytometry returns a lot of data very quickly, which is just a nice feeling sometimes when you've just butted your head up against a wall trying to get your Western blots to work, to be able to turn around and just get good data at the end of it.
Just a follow-up question. Is
Yeah.
that maybe a humidity issue, or is it, I'm just...
Yeah, it was possibly a lot of things. You have to use antibodies for Western blotting, and so there was a chance that our antibodies just got too old. They're really, really finicky. There was issues with membranes. We changed the type of membrane we were using, and so we actually went back and DMART ordered us a whole bunch of the old membrane, and that didn't work either. So we tried a number of things. Humidity would be one possibility, but unfortunately I never did get to the bottom of that.
Any other questions? It does not appear so. So if we could thank Molly one more time. And if you want to ask any questions, feel free to ask. And thank you all for joining us late on this Friday afternoon. Enjoy your weekend.